Hey, welcome back to the third lecture of the nervous system. This lecture is coming to you today on April 5th, just after we finish class. Just going to briefly go over these slides again, just hopefully that this will supplement your learning and you guys will find this material a little bit easier to wrestle with. So we talked about, just to back up, we talked about how the neuron is the functional unit of the nervous system, and we talked about the receptors, which are going to be present within the dendrites, which are going to receive a message, some type of stimulus or input, which is going to come into them. And we said that neurotransmitters were going to be synthesized within the cell body, and then the action potential was going to travel down the axon. We talked about within the axon, there was going to be these areas of myelination. We talked about myelination just being this insulation that covers the axons, which helps speed up conduction. And we talked about action potentials just being this electrical excitability that occurs. Um, Within the action potential, we talked about how the action potential was generated. And we talked about the resting membrane potential being present really around negative 70 megavolts for the neuron. And then we said that whenever the stimulus reached negative 55, then we had something called depolarization, in which the sodium channels were um, primarily responsible. And then once the action potential was generated, it would then begin to come down and become more negative, and we become back down to something called repolarization, in which the potassium channels would be primarily responsible. And then again, we would reach the resting membrane potential. Awesome. So after the action potential has fired, there's a period of time called you guys remember? The refractory period, in which another action potential cannot be generated. We talked about two different types of um, refractory periods. There was one in which absolutely another action potential could not be generated. You guys remember that one? It was the absolute refractory period. Awesome. And then there was another in which a supra threshold or a greater than normal stimulus could generate an action potential, and this was called relative refractory period. Um, so, if you guys remember the type of conduction that occurs in between the fancy word nodes of Ranvier, which is just the area of discontinuous myelination along the axon, do you remember what type of conduction that was called? There's continuous conduction, but which is the one that I'm looking for? Saltatory conduction. Absolutely good. So now we're going to begin talking about factors that affects the speed of conduction. So we said that myelination would be an important component of this conduction, right? So the amount of myelination, that makes sense that the more myelin that you have um, would increase the amount of conduction or the speed at which conduction occurs, right? So myelinated fibers are going to have a quicker conduction than unmyelinated fibers. Another interesting one is temperature. Okay, temperature is going to be interesting. And I'm going to tell you why. There's a condition called multiple sclerosis, right? MS. And this is a autoimmune disorder that causes destruction of the myelin sheath. So you have autoantibodies attacking the myelin present within the neuron. So it can attack everywhere from the central nervous system to the peripheral nervous system. This causes various neurological phenomena, right? This may cause problems with vision, problems with numbness, tingling, can cause motor problems, things like that. So these patients will, whenever they move towards the equator, whenever they move south, their symptoms get better. And I pose the question, why does this happen? Think about it. Well, if you answered because of the temperature change, you would be absolutely correct. It's the temperature change which is going to lessen the symptoms because it's going to increase the speed of conduction, right? We said that an increase in temperature will increase the rate at which conduction occurs, and this is why symptoms get a little bit better because overall, um, the destruction of the myelin causes problems within the nervous system, but increasing the temperature at which the body is living at helps a little bit with the conduction. So that's just kind of a a little tidbit. I think it's cool to kind of relate some of this physiology to real life situations and things. Um, so here we're going to talk a little bit about the speed of impulse propagation. It's important to note that the propagation speed of a nerve impulse is not related to the stimulus strength. Okay, so larger myelinated fibers conduct impulses faster due to the size and saltatory conduction. So it's not necessarily the strength of the stimulus; it's more it's more related to the size of the fiber. Okay, the presence of myelination, that stands for saltatory conduction. Saltatory conduction occurs at the nodes of Ranvier, right? The little areas of myelin uh, present within the axon. And temperature, right? An increase in temperature would affect the speed of impulse propagation. So this just goes to show you that it's not the strength of the stimulus, it's more or less the parameters in which the stimulus is coming to. There are different fiber types, which I'm not too crazy about testing you guys on. There's A, B, and C. A being the largest, right? These are going to be more or less related to skeletal muscle. B, which is going to be your visceral organs and your autonomic preganglion. If you remember, we talked about the autonomic nervous system being in relation to the viscera or your internal organs. And then C is going to be your smallest. These are going to be your sensory and autonomic motor. So I'm not really going to test you guys on these, just letting you know there's different fiber types and different sizes. What is important is knowing the two types of synapses. So if you guys can remember, we talked about the synapse being the communication space between usually two neurons or a neuron and a muscle fiber. There is something called electrical and chemical. Electrical is going to be an electrical signal occurs through gap junctions, and that's going to be important for you guys to know. So, electrical conduction occurs through gap junctions, and this will be gap junctions will be present within smooth muscle. Also, we'll have specialized gap junctions within the heart, in which they will communicate through something called intercalated discs. Hope you guys remember that and are familiar with that from our discussions on tissue. So this is how this is a type of signal transmission. And then there's something called chemical, and chemical is probably going to be something a little bit newer for you guys. You may have not heard of this, but chemical is going to be information transfer from a meaning before the synapse, presynaptic neuron to a postsynaptic neuron. And chemical comes in the form of information transfer in the terms of a neurotransmitter. So a neurotransmitter is going to be the chemical messenger which is responsible for transferring information from the presynaptic neuron to the postsynaptic neuron. And I'll show you guys this in a picture. So this is a type of chemical synapse in which you have a nerve impulse, which is generated down the axon to the presynaptic neuron, and this nerve impulse is going to trigger calcium entry. Okay, calcium entry to the synaptic, or and here to the cell body, and then from here the neurotransmitter is then going to be released. Okay, the neurotransmitters are released, and again, this is the presynaptic neuron. This space in here is called the synapse. Okay. This is the synapse. This is the area between the presynaptic neuron and the postsynaptic neuron. And this chemical messenger is coming from the presynaptic neuron and is going down to here to the postsynaptic neuron. And it's containing a chemical message. It's going to make something occur down here. So this is called a chemical synapse, and I think that it's important to know that neurotransmitters transmit information in the form of chemical messengers from the presynaptic neuron to the postsynaptic neuron, right? This generates a nerve impulse, as you'll see here in number 7. So now we'll read the text and see what the text says. It primarily just says what this picture says, right? Action potentials reach the end bulb and voltage-gated calcium channels open, right? We know that. Calcium flows inward, triggering the release of a neurotransmitter. Awesome. The neurotransmitter crosses the synaptic cleft and binds to ligand-gated receptors. The more neurotransmitter released, the greater the change in the potential of the postsynaptic cell. So you have a nerve impulse coming down here, triggering calcium release. The neurotransmitter diffuses across and binds to the postsynaptic receptor. The postsynaptic receptor then generates a nerve impulse due to the opening of this ligand-gated channel. Again, this is just a picture kind of depicting what we've already discussed in the last. So once we have came down here to this postsynaptic potential, we have two types of potentials that can occur. Something called an excitatory or inhibitory. So an excitatory postsynaptic potential is going to result in depolarization, right? Depolarization, right? It results from the opening of ligand-gated sodium channels, right? So the postsynaptic cell is more likely to reach threshold in order for an impulse to be generated. So again, just to kind of clarify what I previously just said, if the neurotransmitter generates an excitatory postsynaptic potential, an EPSP, right? This is going to result in the opening of sodium channels and depolarization to occur. Okay. Now the opposite of this would be an inhibitory postsynaptic potential, right? This results from opening of ligand-gated chloride or potassium channels, right? It causes the postsynaptic cell to become more negative or hyperpolarized. So again, inhibitory postsynaptic potential results in the opening of potassium channels, which results in hyperpolarization, which just means the cell becomes more negative. So hope that you guys are kind of getting this. Removal of neurotransmitters. Okay, so once the neurotransmitter has came in and once it has been released from the presynaptic neuron and it's diffusing across the synapse and going to the postsynaptic receptor, how do we remove this neurotransmitter from the synapse? The two important ones that I want you to know is enzymatic degradation and the reuptake by neurons. Okay. We talked about acetylcholine, and acetylcholine was released from the presynaptic neuron, and it went down and it bound to muscle fibers and caused muscular contraction. Well, would it be good for acetylcholine to just constantly be present within this synapse? Well, the answer would be no, it wouldn't be, because how would we release more acetylcholine, right? Would we just want the muscle to constantly be contracting? Absolutely not. Enzymatic degradation results in the enzyme being broken down to the choline portion and then the acetyl portion for it to be taken up into the presynaptic neuron. So ACE, ASE, that's present at the end here, lets you know that this is an enzyme, right? And we know that enzymes break down proteins. So this is going to result in the breakdown of acetylcholine. Another interesting one is reuptake by neurons. So again, looking at this picture back here, if you have a neurotransmitter that's going and binding to a postsynaptic neuron, we have something called reuptake proteins that are usually present here. So after it has bound to its receptor, and once it's done its job, we have reuptake proteins which will reuptake this neurotransmitter and take it back in to this back into the presynaptic neuron. So this is actually how SSRIs work, selective serotonin reuptake inhibitors. If you inhibit the reuptake of something, then what happens? If you prevent it from coming back into here and being uptake back into the presynaptic neuron, it stays in the synapse more. And this is how drugs like Prozac work, our selective serotonin reuptake inhibitors. So I want you to know that reuptake and enzymatic degradation are going to be two important means of removal of neurotransmitters. Okay. So once we have a presynaptic neuron generating a neurotransmitter, it binds to the postsynaptic receptor. There are three possible responses that can occur. One, we can generate an impulse, right? If this is an excitatory postsynaptic potential, then the membrane potential has to at least reach -55 for depolarization to occur, right? Or we could result in an inhibitory postsynaptic potential, right? This is in which the membrane is hyperpolarized, right? It's went below the -70 threshold. Or we can generate something called a small excitatory postsynaptic potential, right? If you have multiple small EPSPs, right, excitatory postsynaptic potentials that don't quite reach threshold but are almost there, it can result in depolarization. This is called summation. There's spatial summation and temporal summation, okay? So summation is this: if several presynaptic end bulbs release their neurotransmitter at about the same time, the combined effect may generate a nerve impulse due to summation. So this is whenever you have release of a neurotransmitter, but it doesn't quite reach threshold, right? It doesn't quite trigger depolarization, but we have multiple neurotransmitters that kind of work together, right? It's the sum of these. It's the sum of these added together. This results in generation of a nerve impulse, and this is called summation. It may be spatial or it could be temporal. So, spatial summation is a summation of effects of neurotransmitters released from several end bulbs, but here's the key, onto one neuron, right? Whereas temporal summation is the summation of effect of neurotransmitters released from two or more firings of the same end bulb in rapid succession onto a second neuron, right? So here you have onto one neuron, here you have onto the second neuron. Here's a picture here just kind of showing you how summation works. Here you have one, two, right? So all from the same presynaptic neuron, and then it causes the postsynaptic neuron to eventually reach threshold for the action potential to be generated. So both excitatory and inhibitory neurotransmitters are present in the central nervous system, okay? So you have both excitatory and inhibitory neurotransmitters present in the central nervous system, right? The important neurotransmitters that I want you to know, okay, that will be testable will be glutamate and norepinephrine. So glutamate and norepinephrine are excitatory neurotransmitters. This means they generate depolarization, right, of the postsynaptic neuron. So again, just to reiterate that, glutamate and norepinephrine are important excitatory neurotransmitters. Okay. Now, inhibitory neurotransmission, right? So inhibitory. So remember, these cause hyperpolarization, right? We said that they primarily work through chloride and potassium channels. Okay. So these cause hyperpolarization, in which the neuron becomes more negative than the resting membrane potential at negative 70. So the important one that I want you to know is GABA. This is gamma aminobutyric acid, or short GABA, GABA. So GABA is going to come in different forms, or I'm sorry, there's going to be multiple different ways to stimulate GABA, right? And this could be in the form of a medication called Neurontin or gabapentin, right? This is used for diabetic neuropathy. And alcohol also stimulates GABA. So it kind of slows you down by inhibiting this or causing inhibition on the postsynaptic neuron. Here are the molecular structures of these different neurotransmitters, just to show you that they do have molecular structures. Again, I'm not going to test you over the structure of these, but here is glutamate that's present here. Glutamate is a form of an amino acid. Here are other biogenic amines. There's norepinephrine and epinephrine. These are synthesized in an area of the body called the adrenal medulla, and this is present on the adrenal glands. This is how you get the name adrenaline, right? So these neurotransmitters have different effects, right? Neurotransmitter effects can be modified. So synthesis can be stimulated or inhibited, release can be blocked or enhanced, removal can be stimulated or blocked, receptor site can be blocked or activated. What's important to know is the agonist and antagonist. So an agonist is anything that enhances a neurotransmitter's effect. For example, alcohol primarily works on GABA. Alcohol is an agonist of GABA, right? So it enhances the effect of GABA, resulting in postsynaptic potential inhibition. Does that make sense? Just to repeat that, right? Alcohol primarily Alcohol primarily is an agonist of GABA. So GABA is inhibitory. Whenever you drink alcohol, it works on the GABA receptor and makes it work better. Okay. So an antagonist is anything that blocks the action of a neurotransmitter. Okay. There's multiple different antagonists out there. The one that I like to use is naloxone. So normally, if somebody takes too many pain pills or gets into some fentanyl or whatever it may be, the opiates bind to the opiate receptor. There's something called Narcan, which is going to be an antagonist of the opiate receptor. So Narcan works by making the opiate, it kicks it off of the opiate receptor, right? It antagonizes it or blocks the action of the neurotransmitter. This is what reverses overdoses, right? Here are some other important small molecule neurotransmitters. Acetylcholine, right? We talked about acetylcholine in some of the muscular lectures. So acetylcholines are released by many peripheral nervous systems and some central nervous systems. Do you guys know what the central nervous system is comprised of again? It's the brain and the spinal cord, absolutely. And what about the peripheral nervous system? Right? It's the cranial nerves and the spinal nerves. Awesome. Good job. So acetylcholine is going to work at this neuro, meaning somatic motor neuron, muscular, meaning muscle fiber. So it works in between the somatic motor neuron and the muscle fibers, right? So it works by binding to the neuromuscular junction and causing muscular contraction. So I want you to know that acetylcholine causes muscular contraction, right? It works between the somatic motor neuron and the muscle fiber. There is an enzyme called acetylcholine esterase, right? This ASE, remember it's an enzyme. This is how we remove acetylcholine from the synaptic cleft, is through enzymatic digestion. So some of these amino acids are going to have specific transporters. This is a cool note in here that Valium is a GABA agonist. Okay, Valium is it's a medication called a benzodiazepine, right? Benzo. And Valium, there's many different reasons to prescribe it. Panic disorder is one of those um, clinical implications in which you'll prescribe Valium. It's abused on the street, but it's due to this GABA activity, right? Valium is a GABA agonist. Then this means that it works on the GABA receptor, right? Enhancing its inhibitory effect. Trying to close here soon. This is detailing neural circuits. Neuronal circuits are involved in many important activities: breathing, short-term memory, waking up. This is just a network that contains thousands or even millions of neurons. Okay, so you have an input here. An input goes into these two presynaptic neurons, and then eventually come down onto this circuit here. This is a converging circuit in which there's several different inputs coming into one output. And then this is I don't know how to pronounce that. River, I'm not sure. Essentially, for our purposes, just know that there are different circuits that are involved in many important activities, such as breathing, short-term memory, waking up, things that we do without even thinking about. Plasticity, this is important for regeneration and repair, right? So if the brain becomes injured, we have a way to repair it in some circumstances, right? But anyways, plasticity is maintained throughout life. This is how our brain begins to form these neural circuits and rewire itself. You have sprouting of new dendrites. Remember, dendrites are just those areas that receive input. We have the ability to synthesize new proteins, and we can have changes in the synaptic contacts with other neurons. So we can rewire our neural circuit. This is This is a way in which habits form, is rewiring of the neural circuit. There's limited ability for regeneration or repair. The peripheral nervous system can repair damaged dendrites or axons. This occurs through Schwann cells. So the Schwann cells can help regenerate myelination in certain circumstances in the peripheral nervous system. However, the central nervous system doesn't really, you can't really repair itself. What's kind of cool here is when there is damage to an axon, usually there are changes called chromatolysis, right? There's something that results by usually the third to fifth day degeneration of the distal portion of the neuron, And the myelin sheath, this is called Wallerian degeneration, and I would like for you to know that. Wallerian degeneration is just distal degeneration of the neuronal process, right, in the myelin sheath. Um, Regeneration can occur in certain circumstances following chromatolysis. This all depends on whether the cell body is intact or whether it's um, been lost. Remember, the cell body is going to contain the rough endoplasmic reticulum, which is again going to be needed for protein synthesis and neurotransmitter synthesis. So, again, it's important to know that in order for regeneration to occur, the cell body must be intact. So, axons and dendrites may be repaired if the neuron cell body remains intact and the Schwann cells remain active, right? Schwann cells are going to be needed in order for regeneration of the myelin sheath. So this is a normal neuron in which you have the cell body, you have these dendrites extending out, you have this, remember these are called the nodes of Ranvier in which myelin is covering the axon, right? This is a Schwann cell which is responsible for formation of the myelin sheath. This is called Wallerian degeneration, right? You have lysis of the cell or some type of injury that happens here. And there is distal degeneration of the myelin sheath in the axon, right? This is called Wallerian degeneration. We can have regeneration, right? If the Schwann cells remain active and the cell body is intact. And in this case, it is, right? The cell body's fine, it's not been lost, and the Schwann cells have remained active. So remyelination can occur. So repair within the peripheral nervous system usually occurs by 3 to 5 days, while Wallerian degeneration occurs, which is just breakdown again of the axon in the myelin sheath distal to the injury site. Right within several months, regeneration occurs. The axonal bud grows down the tube to reconnect, and the neural limit on each side of the injury repairs the tube. So this concludes lecture 3 and our nervous tissue lecture. Again, I know this has been difficult to kind of navigate through, but I'm very proud of you guys for making it through these 70 slides. I promise you, wrestle with the material a little bit. Have questions, come to me. Let's talk about it. Let's dive deep into some nervous system. But if you have any questions, please feel free to reach out to me. Thank you guys so much.